I'm John O'Brien. I'm, as you can see there, I'm an old age psychiatrist from Cambridge, and our group has been leading the imaging part of the PREVENT study. So thank you so much, and Craig said how many times we'll say thank you, but you know, all the scans that you've had, and I hope you'll see some of the very exciting data that's emerged from them. And I work with a fantastic team of scientists, many of whom you'll have met here today, who have really done all the hard work in terms of the imaging collation, quality assurance, and the analysis. So you all know why we're doing the PREVENT study and what imaging might be able to contribute. So particularly in regard to brain imaging, we want to get some evidence that midlife is the time at which things are starting to happen, or if not, what is the time things are starting to change? Because the real questions were whether we could demonstrate either structural or functional changes in people who are at risk of future dementia. And if we can, then how are they related to what we might call inherited factors, so genes, but also things that you can't modify like your age, and to what extent they're related to environmental or acquired risk factors. Things, and I'll talk more about these later, but things like smoking, high blood pressure, raised cholesterol, things like that. Because if we understand a bit more about that, we'll have some objective evidence on what we can do to intervene. How do these changes progress over time? That's why we've been very interested in looking at change over time. Are they cause or effect of dementia risk? What do I mean by that? Well, I mean, are these changes, if we see any, are they because there's some early pathology in the brain and they're sort of a sign of that? Or are they actually a very early sign that triggers pathology later on? Because if so, we might be able to alter those changes and intervene. By finding out if and when these changes occur, we can say something about the age, as Craig talked about. Is it 40, is it 50, is it 60, is it earlier? And then particularly in studies of prevention, we are interested in whether any changes we see on imaging can be what we would call an intermediate biomarker for outcome. What do I mean? I mean that if you do a prevention study and people are 20 or 30 years from developing cognitive problems, it's going to be a really long study to show an impact on cognition. If you have an intermediate imaging biomarker, whether that's a structural change or a blood flow change or something else, you might be able to see if some intervention has an impact on that at a much earlier stage. So that's really important information to have. So we've talked a bit about risk, and Craig did as well. So how in PREVENT dementia do we think about risk? Well, as you'll know, the main way people came into the study, and thank you so much for coming in, is because you either had a family history of dementia or you didn't, sort of risk group or a control group. And then once we have been able to look at genes, we can see if people have one of the risk genes for Alzheimer's disease, ApoE4, or not. And about 20% of all of us have this risk gene, so it's quite a common gene. And it's roughly doubled in people who have Alzheimer's disease. So it's a risk gene, it's not a causative gene. But as well as those two ways of looking at risk, so family history or genetics, we can also look at risk in the way of these sort of modifiable risk factors, so particularly things listed there, vascular factors, and the non-modifiable, as I touched on, age, sex, and genes. Non-modifiable at the moment, but of course you may know from other areas of medicine that genetic things can potentially be modified too through various ways in the future. But if we think of them as non-modifiable for now. And one of the ways of bringing together some of these other risk factors is a score that was developed as part of another study called the CAGE score. It's Cardiovascular Aging and Incidence of Dementia score. And it brings together a number of both modifiable and non-modifiable factors. So it brings together age, education, sex, blood pressure, body mass index, cholesterol, physical activity, and also genetics. So it's a kind of compendium score to be able to look at a number of things that are at risk. And one thing I'd just like to highlight here, I suppose, is that in this previous study by Mia Kivipelto, from Finland, she found that a high score, so being above 6, puts you at increased risk of developing cognitive impairment later. And maybe one thing I would just highlight is, you might see here, there is genetics in it, ApoE4, where the laser's not particularly strong. You get 2 points for having an E4. Now, you can't sort of do much about that. That's our genetics. But there's a lot you can do about some of these other things: physical activity, cholesterol, body mass index, blood pressure, possibly even education as well. So that even if you have a gene that puts you at increased risk, you can reduce your risk by getting your score down on these other things. So it's partly, this isn't inevitable at all. Risk, you can modify it. I don't really need to tell you about this. You've all been through the PREVENT study. You know it's pretty comprehensive assessment, and we've asked people to come back at visit 2 to 3 years, and we will be asking, some people have already come back for visit 3, 5 to 8 years. These windows being a little bit wide because of the pandemic, but that's actually, I think, going to be a strength to the study rather than anything else. And we have the 700 in the main study, and then the other 102 and growing contact sports players. And the average age of the main PREVENT cohort is 52. And on average, participants are 24 years away from dementia risk. So this is really going very early, this study, and this, as Craig said, was one of the unique features about it. And obviously the comprehensive clinical assessments, we do the brain imaging, MRI, which we ask everybody if they're able to and don't have contraindications, if they'll be willing to have. And through that, we can look at structure, we can look at vascular changes, things like blood flow. We can look at connectivity, and we can also look at elements of chemistry. And then some of you will have volunteered to take part in some of these other studies. So PET imaging studies, looking at brain amyloid. Stephen Carter there, Stephen, very involved in bringing that data together and looking at that. Tau imaging, Paresh, and these are the tau imaging study which is taking place at Imperial College, so scanning at the Hammersmith Hospital in London. 7T MRI, so this is ultra high field strength. The scans that you would have had if you had a scan would be a 3T, 3 Tesla MRI, which is higher than the standard 1.5 Teslas in most hospitals. 7 Tesla is really pushing things, and you can get much better signal to noise and measure things you can't measure at 3T. And that was combined with virtual reality testing with the headsets. Some of you will have had that. And this was a study done by Coco. Where's Coco? Coco, great. It's just been able to publish those results, so that's really interesting. And if you haven't done the virtual reality task, actually finding your way to the toilets here and back is it's pretty good actually. If you did that okay, you're doing all right. So the virtual reality task is a little bit like that, except you have to kind of pick up flags and stuff on the way. And Coco, you've got a headset, I think, out there for people to try. And Coco will be starting another round of the virtual reality in the new study as well. So one might ask, well, okay, why did we choose MRI imaging for the PREVENT study as our main modality of choice? And partly that was because it's widely available. Now, that meant that in all the 5 sites, we could use the same type of scanner, because all 5 sites had them. But it was more than that, just the scanners were there. It's because if we find something that's relevant, either in terms of early diagnosis or a clinical study, we can generalize these results because every hospital has an MRI scanner. As you know, many of you will have had MRIs for other reasons apart from PREVENT. There's no radiation dose involved, no ionizing radiation, so you can safely have multiple scans. And that's important in a longitudinal study. It provides very high quality structural images, particularly at the 3 Tesla we're using. But not only that, you can image these other things as well, and we'll be looking at some of these: vascular changes, changes in the white matter, metabolic changes, and changes in brain cell density and connectivity. And it's cheap-ish. I mean, the sort of scans in Prevent are £500, £600. It's a lot of money, but it's much less than a PET scan, which would be a couple of £1,000, for example. So it's, and that's the cost for a fairly long scan, as you'll know. How long is the scan going to be? If one focuses down on a clinical MRI scan, you're talking more about £150, £200, something like that. So they're sort of affordable if we find things that can be rolled out. But of course, as you'll know, in the Prevent study, you have a long scan time. That's because we're measuring lots of different things. Clinical scan is much quicker. Enclosed scanner can be a problem for some people. And they are noisy. There's the banging, clanking. Not as bad as the early generation scanners, but nonetheless, they can be noisy. And you might think, well, how does that MRI work? You know, how does it produce a picture of my brain? Well, in contrast to a CT scan, which is sort of shining X-rays into the head and seeing the extent to which they're absorbed and measuring them the other side, and then building a picture of what must have been in the brain to absorb them, this is almost the opposite. You lie in the scanner, you have it has a very strong magnetic field, as you'll know, and allow metal in, and credit cards, these kind of things. So you lie in a strong magnetic field, and what MRI is really doing is looking, in the form we're using, at protons or hydrogen atoms, of which obviously we've got a lot in the brain, in water and in fat and in other tissues as well. So it's really looking at water in the brain, and it's looking at the way that water behaves, the properties of it. So if you're not in a magnetic field, your water molecules, they just kind of diffuse around and they're randomly rotated and spinning as well. But if you go into a magnetic field, they line up in the direction or against the direction of the field. So they're all lining up, and then pulses of radio frequency energy are given which knock these protons out of alignment. So you're actually putting energy into the brain. You're knocking these protons out of alignment, so they're no longer lined up like this. And then they return to line up like that in two sort of different ways. There's a direction of spin, like the sort of north-south pole, the Earth. There's a directionality element, and there's also a rotational element. So to start with, they're all rotating in exactly the same phase, and they start to lose their phase once they're going back to this to align with a magnetic field. And these are two properties of MR that you can measure. They're called the T1 and T2 relaxation times. And because you know where in the brain these signals are coming from, you can then build a picture of the brain back up again. A bit like CT, but so CT is putting energy in through the brain. MRI is putting it in, but what you're actually measuring is that energy being emitted once these protons line up with the magnetic field. And these are the sort of things that we can see after the 80-90 minute scan. We can see good evidence of the structure of the brain. We're looking in some detail at the hippocampus, what we call the subfields. So these are very small parts that make up the hippocampus, which is very much involved in memory and spatial memory. We can see white matter changes, so these white sort of blobs usually represent some early vascular changes. We can do other forms as well that look at evidence of small vascular lesions, small bleeds in the brain. And we can look at the way the brain is connected to each other as well, using connectivity. And other things as well. Arterial spin labeling is a very interesting method that I'll show you some results of later, where you actually magnetize protons in the blood before they go into the brain, and then you actually measure that loss of magnetization as they come into the brain, and you can measure blood flow using that method. And that's been very, very exciting in the Prevent study. Okay, so what have we found over the years? So this was actually the first paper we published. Jamie Stefaniak who was a young scientist with us at the time. And just to echo what Craig said, this has been so important in training a whole new generation of researchers, as well as finding some interesting things. We knew that there is a strong relationship between vascular brain changes and dementia. They're more common in people who have dementia, including Alzheimer's disease. And we know that they contribute to some of the cognitive problems people with Alzheimer's disease get. And they may even increase the risk of getting Alzheimer's disease. So we asked a very simple question. Did people in the PREVENT study in midlife, visit 1, did they already have evidence of increased vascular changes in the brain if they were at increased risk of dementia? Because if so, you might say, well, you've already got brain changes there, you've come in a little bit too late. What we found was the opposite, actually, which was that we didn't see any increase in vascular changes in people who were at increased risk of dementia, whether we looked at that by virtue of family history or the ApoE4 gene. On a number of different markers, so white matter lesions, small black blobs, infarcts. However we looked, there was no particular difference. So that's very reassuring. That suggested that at the mean age of 52, the brains were well preserved from a vascular point of view. Marilina Dunavi, where's Marilina? Yes, over there. Marilina looked in greater detail at the brain structure, so moving on from vascular changes. She looked at the general brain structure and again found that the brains were very well preserved. There wasn't evidence of volume loss or atrophy in most of the brain in people who are at increased risk. What she did find, though, is that if you delve down into the very inner depths of the hippocampus, into certain cell layers, the details aren't particularly important, but one of them is quite interesting because it's where you get early deposition of the tau protein, which along with amyloid, as you'll know, I'm sure, is the hallmarks of Alzheimer's. We saw some very subtle changes at a group level in people who are at increased risk. So this was the only area of the brain we saw any difference in these small cell layers in the hippocampus, in the absence of any change in whole hippocampal volume or whole brain volume or anything else. So our conclusion from that was that maybe these changes were just beginning. These subtle changes were just starting to happen. Marilina went on to look in more detail at the brain structure in other ways, looking at the thickness of the cortex, cortical thickness, and again found that there were no differences between risk groups except in looking at this CADE score. So these is the sort of environmental factors, the vascular factors, things like that, and did find that people with a high CADE score had a significantly thinner cortex. And this was a sort of common finding in our work, that we found some of these vascular risk factors and other risk factors seem to be associated with structural change in a way that the genetic and other risk factors weren't. Then Liz McKernan, who I don't think Liz is here today, but Liz did studies with Marilina on blood flow. So this is using this arterial spin labeling method that I mentioned, and found, and I think there was a question about unexpected findings. This was probably one big unexpected finding for us, that blood flow was increased in people who were at increased risk of future dementia. So if you had a family history of dementia or you had the ApoE4 gene, you had a higher blood flow in your brain than people that didn't. And this was quite surprising because when people develop cognitive impairment, their blood flow goes down because there's not so much brain working properly to use the blood. But here we saw an increase, and a very significant increase. You can see widespread, all those coloured areas, a significant increase. And so that led to a new series of studies I'll talk to, but it's just very interesting that we don't quite know the cause of that, and we want to look into that in more detail, but it's an early change. It's an early change, and it's either a response to something that's going on in the brain already, or else it's something that might be causing a problem later. And that's really important to tease out, because if it's the latter, if it's causing a problem later, we'll be able to do something about that and intervene. And then Marylene looked in more detail about this and suggested that it was probably due to some dysregulation of the normal system by which blood flow is controlled. And ApoE4 has a number of functions which include an effect on the vasculature. Audrey Lowe, Audrey? Where's Audrey? Audrey over there, yep, thanks, has been looking in more detail at some of the modifiable risk factors, and in terms of vascular changes in more detail than we were able to before, and also to look at change over time. So again, this is looking at things like these white matter lesions, these white blobs, looking at small areas of infarcts and small areas of bleed, and looked at people with, in regard to these modifiable and non-modifiable factors, and found, again, really supporting evidence that it's the modifiable factors, so things like high blood pressure, high alcohol, increased weight, head injury, which Willie will talk about that later, and hearing loss. They're all potentially modifiable, at least, and they were the factors that were associated with small vessel disease, rather than the sort of genetic or other factors. And I think that's a really, really important message, because these are things that we can do something about, people can do something about. Elijah Mack. Elijah, great. So, as you can see, there's a number of great people have been working on this data. Elijah's been interested in looking at another imaging method which is able to look at the density of nerve cell connections in the brain, and called dendritic density, using MRI imaging in the grey matter. So this is the sort of richness of connections that brain's nerve cells make to each other, if you like. And in doing this study, we also collaborated with another big study in Spain, in Barcelona, called the ALPHA study, which stands for Alzheimer's and Families. And it's quite a similar study to PREVENT, in that it's got people who have increased risk as a family history and those who don't. But the participants are older than PREVENT participants by about 10 years. But putting the two studies together, we could get an enormous sample size, almost 2,000 people, which is amazing for an imaging study. It really, really is. And it gives such huge power. And what Elijah was able to do, using this method of looking at the kind of complexity of nerve cell connections, was show an effect of ApoE4 and in relation to age. And these curves might look sort of fairly similar here, but what this shows is the sort of difference between people with and without the gene in terms of their dendritic density, if you like, over time. And as Craig mentioned earlier in life, if anything, ApoE4 is good for you. It seems to have a lot of benefits in cognition and also in terms of brain structure. But beyond a certain age, it becomes more problematic. And Elijah was able to look at that sort of age at which there seemed to be an impact of E4 in changing this complexity of nerve cell connections, and found that it was around the ages of sort of 63 to 70, so later in life than we were seeing the impact of the more modifiable risk factors, almost as if the genetic factors are kicking in a little bit later. And we also looked in terms of looking at 2 scans done from visit 1 to visit 2, and looking at change over time in the rate of brain volume change, if you like. And again, we found a similar message, that there wasn't anything in terms of ApoE or family history, but again, it was this CADE score, these kind of vascular risk factors that seemed to be associated with increased volume loss. And if you had a high CADE score, over 6, which was the number I mentioned before, your risk of brain volume loss over 2 years was much higher than if you had a lower CADE score. So it's really, the message from all these studies was very, very consistent about these sort of vascular modifiable risk factors. Another study from Audrey, I'm just in the interest of time going to skip over that and talk about where we want to go next with regard to some of these studies. And again, thank you to the Alzheimer's Society, but they've recently funded a fellowship to Marolina Dunavi to undertake a new study called the Charisma Study, which she'd be very happy to talk to you about. You need a good acronym for study, and you can see how she came to her acronym. Cerebral hemodynamic analysis in relation to risk factors for mild cognitive impairment in Alzheimer's disease. It's quite a mouthful. Charisma's great. So the Charisma Study, and what Marolina wants to do is to follow up these changes that we found in terms of blood flow, and to look in much more detail at why these are occurring. We did sort of what you might think of as a fairly basic sequence to look at blood flow. We can do much more detailed sequences to look at the blood vessels, to look at whether they're pulsating normally, flow in different parts of the brain, and various other things as well. And her study, which starts next month, will be asking for volunteers to come back and have this extra MRI scan so we can look at this vasculature in much, much more detail to try and work out what's going on. And as a part of this study, we're introducing a relatively new imaging technique, which is called near-infrared spectroscopy, which is literally shining light into the brain. So this is a form of brain imaging, and Amelia Buttons is a student who's working on this. And that's because some type of light, particularly the sort of infrared light, it can actually travel through the skull into the brain and come out again and be detected. And through that method, you can actually measure the extent to which the blood is oxygenated and the extent to which it isn't. And that will give you a kind of bit of a handle on how well the brain is working, whether its metabolism is normal. And the thing about the NIRS, there's a NIRS cap you'll be able to have a look at, is that it's not an invasive procedure. You have to wear a cap, but you don't have to come into a scanner. You can have your NIRS scan sitting at your computer or whatever. So this can be done in other settings, can be done at home. You can have a NIRS scan walking down the street, if you don't mind looking like that when you're walking down the street. And Marolina's study is combining MRI with NIRS and probably your favorite cognitive test, the Four Mountains test, which defeats me. Anyway, there we go. So just in summary, what we've found from PREVENT is that people in midlife at risk of future dementia have generally preserved brain structure and no evidence of vascular damage, but subtle changes are starting to be seen in some areas. And we do see increased blood flow. Over 2 years, as we followed the cohort over time, we are starting to see some atrophy and white matter lesion progression. There is some interaction with E4, but the main effect is actually on the sort of modifiable risk factors. And it does suggest that I think PREVENT is probably the only cohort that's really produced this objective evidence of time to intervene. That's been really, really important, is the appropriate time for dementia prevention. So take-home messages, I'd say, judging by our imaging studies, your brains are in pretty good shape. So that's good, I'm pleased. Looking after your vascular health will look after your brain. Don't worry too much about your risk and your genes at the moment. Think about these other factors. Consider getting some help to stop smoking if you smoke. When I say drink in moderation, I always feel I have to say, if you don't drink, it doesn't mean you should start, you know. If you drink mildly, it doesn't mean you should double your amount of alcohol. Just don't drink too much, I suppose. Take exercise and eat well. Thank you all so much for volunteering your valuable time and also your patience in having all these scans. I know they sometimes can be cancelled, things can sometimes not work. You've been absolutely fantastic in having them done. We'd still like you to have more though. And you've probably met the team, but please do meet the team at the exhibit stands. Happy to answer any questions before lunch. Thanks.